In this practical, we learn how to prepare cultures of microorganisms using aseptic technique and then use those cultures to test disinfectants and antibiotics by measuring zones of inhibition. Sterile nutrient agar in petri dishes and sterile broth provide the culture media, and we label a base with name, date, and organism and incubate plates upside down at about 25 degrees Celsius in school laboratories. We work with a sterile inoculating loop or a sterile spreader beside a lit Bunsen burner, and we begin by tying back hair, washing hands, disinfecting the bench, and keeping talking and movement to a minimum. We sterilize the loop in the blue flame until it is red hot, then let it cool without touching anything, and we flame the neck of culture bottles before and after opening. For a streak plate to obtain single colonies, we open the lid like a hinge, touch the cooled loop to the culture, and streak across 4 zones, reflaming and cooling between zones to dilute the cells. For a lawn culture, we pipette a small volume onto the agar and use a sterile spreader to create an even bacterial lawn suitable for drug sensitivity testing. We replace the lid quickly, tape it with 2 or 3 small pieces without sealing all the way around, incubate the plate upside down, and keep the temperature at the stated safe value. After incubation, we examine the plate and select isolated colonies from a streak plate to subculture and create a pure culture, if that is the goal. To test chemicals, we place sterile paper discs on a lawn plate with discs soaked in different disinfectants or antibiotics at known concentrations and 1 control disc soaked in sterile water. After incubation, clear zones around discs indicate growth inhibition, and we measure the diameter of each zone in millimeters and record the values because a larger zone means more effective at that concentration. We interpret results by comparing to the control, repeating for reliability, and keeping variables controlled such as lawn density, incubation time, temperature, and disc concentration. And we do not open plates after incubation, and we dispose of them in autoclave bags. Aseptic technique protects both the cultures and the people, so we keep plates sealed and inverted, avoid high incubation temperatures, wash hands, and disinfect the bench when finished.